Well, it's absolutely fundamental, because the fly was the instrument by which we discovered the detail of genetics. So Mendel's work, which was rediscovered at the beginning of the 20th century, was clearly extremely important, but on the other hand it wasn't completely convincing, and it's very telling that one of the people who didn't convince was Morgan. So Morgan, when he started using Drosophila, he didn't believe in genetics. He thought it was not, well, the word and the rediscovery had only, the word had come back in 1906, rediscovery in 1903, so Morgan starts using Drosophila in about 1907, 1908, primarily interested in studying evolution. That's what he wanted to look at, so he decides he's going to subject all his flies to all this environmental stress, which he thought, following De Vries, that there would be these mutational periods and you get new forms arising because of environmental stress. And what he then does from 1910 to kind of 20, with Sturtevant and Muller, basically they discover genetics, or rather they forge genetics as it is now known. They show that genes are on chromosomes, and they discover all the various tools you can use with Drosophila. So without that, we would have done something, certainly, but it would have been much more difficult, which is why they chose Drosophila, because it's a short generation time. So I think firstly the discovery of genetics would have been utterly altered, would have been without Drosophila. Secondly, the new synthesis of evolutionary biology, which was to fuse our understanding, the classic understanding of evolutionary biology with the new science of genetics, that required and used Drosophila in an enormous way. And then there's kind of a fallow period for much of the 20th century in which Drosophila drifted off the agenda once the new synthesis had happened, once the turn to molecular genetics happened in the post-war period and the discovery of DNA. Drosophila was too complicated. So in fact what you needed then was more simple organisms, or things that weren't organisms at all, like phage. So Drosophila was very much a backwater. When I moved into it, although it was seen as having huge potential, it was still, it was potential rather than reality. So it was only with the relative ease of gene sequencing in the late 80s that, in fact, it started to become incredibly important again. And also with the realization that there were fundamental developmental genetic characters that turned out, in fact, to be present not only in Drosophila but in all multicellular animals. That then gave it a new kind of impetus. So you got the development of developmental genetics, which was really incredibly important, because in the 19th century and prior to then, the genes, genetics and development, so heredity and development, had been fused in one thing. This idea of generation, this was where the organisms came into being. And then for much of the 20th century we all focused on genetics, on simply the transmission of those characters, and the importance of development kind of got forgotten and was almost seen as being this, it was simply an unfolding, it was simply a readout of the gene, so very, very deterministic, essentially the kind of pre-formationist view of the gene, of what genes were, that they were a blueprint, you could go from one to the other. And with the rediscovery of the importance of development in the work that in the end led to the Nobel Prize, then that showed that in fact development was actually a formative process and was an interaction with the environment, and plus all of these common features with all these with other organisms. So Drosophila did that. It was able to pull two bits of science that had been separated, put them back together and show their importance. And also what particularly interested me, and what got me first interested in Drosophila, was the neurogenetics, that you could study surprisingly, amazingly, nobody believed them, you could study single gene mutations which would have massive effects on behaviour, be they on learning, which is what got me interested, or in circadian rhythms, whereby a single gene mutation can alter the rhythm of how a fly wakes up, and it's basically the same in the human being as well. So those, I think those kind of key, that's what we see carrying on for the last kind of 20 or 30 years now. Drosophila is absolutely massive field. When I started in the mid-70s, it was more a potential, because we didn't have access to the genome. I think, when I started, no gene had been sequenced, so even the existence of the molecular basis of genetics had yet to be properly demonstrated in eukaryotes. It had all been shown in phage and in bacteria, so it was just kind of taken on as read that this must be the case in the other organisms. But once the molecular revolution really got going in the second half of the 70s and the 80s, then very rapidly people turned back to Drosophila and realised, okay, we can do some great things with this. That was a very long answer.